quickly go across this gated thing. So facilitated diffusion, as I said, yeah, facilitated diffusion is which it is the thing which is taking place through proteins and assemblies of proteins embedded in plasma membrane. So what is basically this facilitated diffusion is transmembrane protein or transporters. They form water-filled channels through which ions and molecules go down concentration gradient means it is from high concentration to low concentration. So this transmembrane channels which forms this water filled channels, and transmembrane proteins which forms this water filled channels. These permit facilitated diffusion that can open and close. Okay, so they are called gated. We have different types of gated ion channels. First one is ligand gated. So as the name suggests, ligand is the one which is gating this thing. So ligand gated, many ions channels they open and close in response to binding a small signaling molecule. This signaling molecule, that is what ligand means. When this small ions which are needed to be transported, when they bind to a small molecule or ligands, they open and close channel in response, that the channels, transmembrane channels, are opened and closed accordingly. Okay, so some ion channels are gated by extracellular ligands, some are by intracellular ligands. So in both cases, the ligand is not the substance that is transported with the channel. So we have external ligand as well as internal ligand. So first we will see external ligand. External ligand means this signaling molecule, small signaling molecules to which the ions or molecules that is needed to be transported as binding is present at the external site, extracellular site of channel. Okay, that is bind to site on extracellular side of channel. We have to see the examples of acetylcholine and GABA. So acetylcholine is a binding of neurotransmitter acetylcholine. It is a neurotransmitter which is acetylcholine. This is at certain synapse about opens the channel that permits sodium ion and initiate a nerve impulse or muscle contraction. Okay, this is the structure. As of structure, you know what is acetylcholine, I think. So we have a carbonyl group with this is actually an ester group. So carbonyl group with the methyl and O, then with the CH2 we have N with the 3 CH3s, that is NCH3 thrice CH2 CH2 O COCH3. Okay, that is our acetylcholine. So this is a binding of this particular neurotransmitter acetylcholine at a certain synapse, so that what binding to this acetylcholine at certain things opens channel for sodium ions, which initiates the nerve impulse or muscle contraction. Now next we have GABA. GABA is nothing but gamma aminobutyric acid. So binding of GABA at certain synapse designated like gamma A in central nervous system admits these chlorine ions into cell and inhibits creation of nerve impulse. So GABA is also an external ligand. So binding to this GABA at certain designated synapse means where it is needed to be at particular point. Okay, in central nervous system admits this chlorine ions into cell and inhibits the creation of nerve impulse. Okay, these are the two important external ligands. Now we have internal ligands. So internal ligands, as it is saying, bind to site on channel in which is imposed to cytosol. Okay, bind to site of channel protein imposed to cytosol. Then secondary messengers like cyclic AMP and cyclic GMP we have. These regulate channels involved in the initiation of impulse in neurons responding to order and light. So see cyclic AMP, which is cyclic adenine monophosphate, this responds to order, and cyclic GMP responds to light. So these are the channels involved in the initiation of impulse in neurons. Okay. Next, we'll see ATP is needed. Yeah. So ATP is actually needed to open and close these channels. But the point is, the hydrolysis of ATP regenerates this energy for opening of such channels. But this is not an example of active transport, because ions diffuses through open channels following their concentration gradient. So this can't be a active transport. Okay. Next we have mechanically gated, mechanical like sound waves bending this cilia like projections on ear or cells of inner ear, and this open up channels leading to the creation of nerve impulse that brain interpret as sound. Then we have voltage gated in excitable cells like neurons and muscle cells. Some channels are open and closed in response to the change in charge across the plasma membrane. So as impulse passes down the neuron, reduction in voltage opens this sodium ion in adjacent position of membrane. Yes. So this is about diffusion of ions. Now we will see diffusion of molecules. So some small hydrophilic molecules like sugar can pass across this molecules. Now till now we have seen ions, acetylcholine for sodium ions, then GABA for chlorine minus ions, then this thing secondary messengers like cyclic AMP and cyclic GMP. These are regulated. These are involved in the initiation of impulse in neurons corresponding to the smell and odor. So these all are ions, right? Ions is related. So in case of mechanical also we have seen this for opening, sound waves can bend the cilia like projections in our ears, and it can open channel for creation of nerve impulse, that is leading to creation of nerve impulse. Also in voltage we have seen sodium ion channel, that is reduction in voltage opens the sodium ion channel. Yes. Now we will see molecules like sugar and sugar-like molecules also should be passed, right? So some small hydrophilic molecules like sugar can also pass by facilitated diffusion. So this process, of course, it requires a transmembrane protein or a transporter. So in case of ion-filled channels, they form what? Water-filled pores, along water-filled channels, and that enables molecules to pass down its concentration gradient. So we have maltodextrin. Maltodextrin is an example for it is a homotrimer in the outer membrane of E coil. This forms pores that allow disaccharide maltose, that is our sugar, and few related molecules to diffuse into the cell. Actually we are not saying they form water filled pores, they actually form pores. Maltodextrin is actually a homotrimer which forms a pore. This is present in E coil, okay? This forms a pore and which allows this disaccharide maltose and few other molecules to transport. So in plasma membrane we know hemoglobin, right? Yes. So in plasma membrane of human RBC, it contains transmembrane protein that permits diffusion of glucose. Hemoglobin is for diffusion of oxygen. So diffusion of glucose, we have transmembrane proteins that permits the diffusion of glucose from blood to cell. This is the case of facilitated diffusion of molecules. Also, some molecules can pass through membranes by configurational change, in shape of transmembrane protein which binds the molecules to be transported. So also in either cases, interaction between molecules being transported and its transporter resembles in many ways interaction between an enzyme and a substrate. Okay, now we'll see the important topic, active transport. So we know what is active transport, right? It is the pumping of molecule